Distinguished guests, ladies and gentlemen, online viewers worldwide, welcome to the 2016 Tang Prize Award announcements. From June 18th to June 21st, the Tang Prize Foundation will be announcing the laureates of the 2016 Tang Prize in Sustainable Development, Biopharmaceutical Science, Sinology, and Rule of Law. In today's press conference, the 2016 Tang Prize in Biopharmaceutical Science will be unveiled. First of all, it gives me great pleasure to introduce to you the distinguished guests who have graced us with their presence this morning. Chairman of the Tang Prize Selection Committee, 1986 Nobel Laureate in Chemistry and former President of Academia Sinica, Dr. Li Yuanzhe. Vice President of Academia Sinica, Dr. Andrew Huijun Wang. Academician of Academia Sinica and Chairman of Taipei Medical University, Dr. Zhang Wenchang. Academician of Academia Sinica and former President and Distinguished Investigator of National Health Institute, Dr. Gong Xingjian. Board member of the Tang Prize Foundation and President of Taipei Medical University, Dr. Yan Yun. CEO of the Tang Prize Foundation, Dr. Chen Zhenchuan. Welcome. Ladies and gentlemen, it is my honor to introduce to you the chair of the Tang Prize Selection Committee, Dr. Li Yuanzhe, to make the 2016 Tang Prize Award announcement in Biopharmaceutical Science. Dr. Li.
Distinguished guests, members of media, I am now honored to announce the recipients of 2016 Tang Prize in Biopharmaceutical Science. Emmanuel Charpentier, Jennifer A. Dauda, Feng Tang, for the development of CRISPR/Cas9 as a breakthrough genome editing platform that promises to revolutionize biomedical research and disease treatment. I want to congratulate all three of them. Thank you.
Thank you, Dr. Li. And ladies and gentlemen, please join me in welcoming Academician of Academia Sinica, Dr. Gong Xingjian, to introduce to us the Tang Prize laureates in biopharmaceutical science and also to give the award citation.
With great pleasure, I'd like to introduce the winners of this year's Tang Prize in biopharmaceutical science. And I'd like to perhaps briefly first introduce the human genome to you before I talk about their contributions to this genome editing platform. Our human genome is a blueprint of life, and the platform CRISPR-Cas9 is a revolutionary platform for genome editing. If we look at our human genome are strings of DNA with about 3 billion nucleotides or base pairs and about 25,000 genes. Under the electron microscope, you can see that the DNA, the genome, really looks like beads on a string or a very, very long string of pearls. Of course, the genome is a molecular basis of life and heredity. It dictates our physical appearance, our emotions, as well as our health. Defects in structure and expression of genes are often causes of diseases. Precise and efficient platforms to manipulate or edit individual genes are keys to our understanding of gene functions and disease treatment. It has always been the dream of the scientists that someday we can effectively editing human genome for medical benefits. Because of the brilliant work of the recipients of this year's Tang Prize, that someday is today. Now I'd like to introduce the platform to you, the CRISPR-Cas9 platform. It's a little bit complicated. CRISPR stands for clustered, regularly spaced, interspaced short palindromic repeats, and the Cas9 represents CRISPR-associated protein 9. Cas9 itself is a nuclease which cuts DNA, but it was originally derived from bacteria. It's a bacterial adaptive immune system evolved to cut foreign DNA like phage, invading phage DNA for self-defense. But very interestingly, this nuclease, unlike others, requires cofactor, and the RNAs are the cofactors. There are two pieces, CRISPR RNA and the transactivating CRISPR RNA, which can be combined, as Dr. Dudna and Dr. Charpentier demonstrated in a seminal paper, into one single RNA called the sgRNA. And this is shown in the blue line here. This sgRNA can then guide the Cas9 when it binds to Cas9 and can search for the phage DNA. If this is invading phage DNA, can search for the sequence that can match. Once it finds the sequence, the Cas9 can then give a cut, and that's how one can get rid of the phage DNA. Dr. Zhang's work demonstrates that Cas9 not only works in bacteria, but also works in mammalian cells and in human genome. That really opens the gate for human genome editing. Now, the Cas9 is a little bit complicated, so if I may, I'd like to use a simple illustration. If we think about the human DNA like a very, very long string of pearls, there are about 3 billion such pearls. If there's a defect in one of the pearls, it's extremely difficult to identify it and to repair it. And that was the challenge, or has been the challenge, that we face to fix a single gene in the midst of this huge number of other genes and to rid of the disease. But luckily, we have the Cas9 system. We can actually consider the Cas9 as a GPS-guided scissors. Cas9 itself is a scissor that can cut DNA. But the single guide RNA, sgRNA, this is actually the GPS system. When we use the GPS system, we like to input an address. And the GPS system then can use this address to take us to where the particular site we want to go. And that's exactly what Cas9 can do. This single guide RNA can be custom designed, can be custom made. And if we input the DNA sequence of the gene of interest, then this can really target that particular gene. So if you put sgRNA and Cas9 together into human cells, then this sgRNA will begin to search along the human genome. And once they find the match, the sequence that can match, it will ask the Cas9 to give a cut, give a precise cut. No other genes are affected. And that's why it's so precise, so efficient. And then the editing can be performed later. So this is a remarkable technology with precision and efficiency. Next. So now let's introduce the 3 recipients with respect to their contribution to this platform, this technology. Dr. Charpentier is presently director of Department of Regulation for Infectious Biology, Max Planck Institute. She is a microbiologist, well recognized as a world-leading expert in regulatory mechanisms underlying processes of infection and immunity in bacterial pathogens. She made several contributions to the CRISPR field. But if today we have to single out one, that would be the original discoverer that Cas9's activity requires transactivating RNA. So basically, Dr. Charpentier discovered the GPS system for Cas9. Dr. Jennifer Doudna is professor of chemistry and molecular and cell biology at UC Berkeley. She is a renowned RNA structural biologist working on ribosome or other RNA-related enzymes. She, in collaboration with Dr. Charpentier, was credited for the discovery that CRISPR RNA and the transactivation RNA can be fused together into a single guide RNA. And this is very important. It significantly simplifies the procedure so that the whole operation can be so user-friendly. Dr. Fong Zhang Fong, is presently investigator at the McGovern Institute for Brain Research and the core member of the Broad Institute and Associate Professor of Brain and Cognitive Sciences at MIT. He is a bioengineer with interest in developing tools for investigating neuroscience and human diseases. His critical role in CRISPR technology was the first demonstration and adaptation that it can happen in human cells. And that was also a breakthrough development. If we look at the CRISPR already has huge impact in scientific and the medical community. This is reflected by the publications that has accumulated in recent years. In 2010, there were fewer than 50 publications. 2012 and 2013, the recipients of this year's Tang Prize published 2 seminal papers. And then in 2015, we saw that the number of publications rose to about 1,400. So you can see there has been an explosion of interest in the utilization of this technology. So finally, I'd just like to mention a few applications that we can think about, and there are many of them. The first, CRISPR can be used to repair defective genes in a large number of heritable diseases, such as sickle cell anemia. Second, it can be used to fix mutated genes that drive cancer development, such as KRAS oncogene and the p53 tumor suppressor. Third, we can also use it to clear the integrated latent viral genome from the infected cells, such as HIV genome. We can completely get rid of it. And there are many, many other applications. The opportunities are enormous, and they are limited only by our imaginations. Many believe that this CRISPR technology is one of the greatest discoveries in genomic research, rivaling molecular cloning and the PCR. So for this really breakthrough, wonderful accomplishment today, we recognize and congratulate Dr. Charpentier, Dr. Doudna, and Dr. Zhang. Thank you very much.
Thank you, Dr. Gong, and once again, congratulations to the 3 award recipients in biopharmaceutical science. Ladies and gentlemen, thank you for joining us at the 2016 Tang Prize Award announcement in biopharmaceutical science. All of the press information will be made available on the Tang Prize official website. The Tang Prize Award ceremony will take place on September 25th this year. We look forward to seeing you then. Lastly, please do join us from June 20th to 21st for the award announcements in Sinology and also Rule of Law. Thank you very much, and we hope to see you again tomorrow.